Doing Biology Part 2 of May-June 2018, Paper 4.1. Okay, so today we're moving on straight to question number 3. We've done in Part 1, we did question 1, question 2. Today we're looking at question 3. We're starting from question 3. Is that okay? So without much ado, let's begin. He said a student cut a section of a root and made an outline drawing of the distribution of tissues as shown in figure 3.1. Okay, the first question says identify the position of the xylem tissue by drawing a label line and the letter X on figure 3.1. Identify the position of the xylem tissue. Okay, identify the position of the xylem tissue. Okay, and show it with X. Okay. Okay, so what we do, just from here, from this center, this star, just because this is xylem tissue, just draw with an arrow, mark X. Okay, just show it with an arrow, then you mark X. That's all you need to do. From that center star, you show with an arrow. Okay. Like this. Okay. So we just show, let's say this is the okay, let's say this is the structure. The inner has a star, something like this. Okay, so the xylem, this is the xylem tissue. So you just show with X. Question 4a. Okay? Something like this. This is what you need to show for this structure. Okay, so that's that for that. Let's move on quickly. The next question is a state why xylem is a tissue. Why is xylem a tissue? Okay, because a tissue is made up of group of cells, okay, that are similar in structure and doing the same thing. So xylem is a tissue because it's made up of, if you look at it, made up of is a group of cells. So we say group of cells. B. Xylem is a tissue because it's a group of cells. That word is very important. Group of cells, because tissues are group of cells. Okay, that are similar in structure and in characteristics. Group of cells doing a similar function. Group of cells doing the same structure function. Group of cells that are similar in structure. That are similar. Okay, use the word similar or same, and does the same function. Does same function. So function, similar structure. So when you mention similar structure, you get a point, and you mention the function, you get a point. Okay. So a group of cells that have similar structure and does the same function. Same function. So that's tissue for you. So when something is a tissue, it means it's made up of a group of cells. Something is an organ, it's made up of a group of tissues that have similar structure and does the same function. Something is a system made up of is a group of organs that does the same thing. Is that okay? So that's that for that. We move to the next question as fast as we can. So you get your 2 marks. It's a water we see right now. Oh, okay, now we're doing B. The one we did just now was A Roman figure 2, not B. The one we did just now is A Roman figure 2. So this is A Roman figure 2. Now we're doing B. Now we're in question 1B. Now let's take a look at B. Is it water absorbed by the roots move through the stems and enter the leaves? Most of this water is lost in transpiration. Explain how the internal structure of the leaves results in the loss of large quantity of water in the transpiration. Explain how the internal structure of the leaves results in the loss of large quantity of water in transpiration. Okay, now let's take a look at this internal structure of the leaf and its role it played in the loss of water during transpiration. Okay, first of all, we say the leaves, they have large internal surface area. Okay, their body is reconnecting, so we need to wait for it. The internal structure of the leaf is that it's composed of large surface area. The surface area of the leaf is large enough, and then which holds a lot of air spaces, and when the xylem supplies the water during when it's heated up from the heat from the sun, the water evaporates from the surface of the mesophyll layer or the mesophyll cell. The water will evaporate from the surface of the mesophyll. Okay, first of all, we start by saying, okay, our body is laggy, it's not kinetic. We start by saying that the internal structure of the leaf is made up of wide surface area, and we say the xylem cells brings water from the root to the leaves. The xylem cells carry water, transport water from the root to the leaves. And this water, when it gets to the leaf, because of the wide surface area, the large surface area that is available, then this water, when the sun hits the leaves from the top, this water becomes almost at the vapor state, not longer water at the normal water, it becomes water at boiling point. And this water becomes vapor, and this water begins to evaporate through the Gall cells and through the stomata, through the Gall cells and through the stomata. So the water vapor now diffuses, because it's no longer water, it's a steam. This water vapor now diffuses out of the stomata. So the body is not allowing us to write it, but these are the points. Okay? Okay, let's move to the next question. So you get 3 marks for this. You get 3 marks. What I said, you first of all, you measure the internal structure. You say the leaf, the internal structure of the leaf, it has a very large surface area. And then you write large surface area. The leaf is comprised of large surface area. That's your first point. Your second point is that the xylem cells transport the water from the root hair or from the roots of the leaves, from the root of the plant to the leaves, right? This water, on getting to the leaves, is being heated by the sun, okay, by the heat from the sun, and the water gets to changes from water to vapor, and then this vapor diffuses through the Gall cells and the stomata into the atmosphere. So this water is lodged in large quantity because of high temperature. This water is lodged in high quantity because of large temperature that heat the leaves, and the water evaporates from the mesophyll layer, the mesophyll cells of the leaf. Okay, so these are the points you write, you get your 3 marks. These are the points you write, you get your 3 marks. If the board allows or is still connecting, I wanted to give the point. First point is large surface area of the leaf. First point is large surface area of the leaf. Second point is that xylem cells transport the water from the roots of the leaves, the root, I always say the root of the leaf, the root of the plants to the leaves of the plants, okay? And then after that, we say this water get heated up from the heat in the atmosphere, from the heat of the sun in the environment, heats the water up, and the water changes to vapor. And this water vapor evaporates from the mesophyll cells of the plant Okay, so I think the board is okay now. First is, we say, is large surface area of the leaf. Large surface area. Large surface area. Large surface area. So the leaf have a large surface area. That's I will just summarize it like, okay, so here the next point is that xylem will transport the water to the leaf. Xylem versus transport. So I just write only xylem. In the exam you can write the xylem cells or xylem vessels. Transport H2O, which is water. Transport, in the exam write water, not the chemical compound, the chemical formula. Transport water to the leaf, to the leaves. Leaves, I write plural, leaves. Plural of leaf. Okay, then after that we say heated water changes to vapor. You know the heat is coming from the sun, right? Heated water changes to vapor. Changes to vapor. You can say large quantity. You can say large quantity. Changes to vapor. Now the vapor diffuses in the mesophyll cells. Diffuses in the mesophilia or mesophyll cells of the leaf. Mesophyll cells of the leaf. Through, continue the sentence, through the guard cells, or you make this one a new point. Through the guard cells. Through the guard cells, stomata, into the atmosphere. To the atmosphere. Okay, so that's that for this question, and then we'll move on to the next question. Okay, I think we finished question 3. Right? Now we're in question 4. Question 3 was fast, right? Now we'll go to question 4. Question 5 is very plenty. It's very a lot. Even question 4. 4 is a lot. 4 is like 3 times question 3. 4 is a lot. Okay, let's deal with question 4. Now, question 4, it said the flow of blood through the skin can be investigated by using a flow meter. Now figure 4.1 shows a flow mirror above a section through the skin. Figure 4.1 shows a flow mirror. This is the flow mirror. Above this is the skin. See, figure 4.1 shows a flow mirror above a section through the skin. Okay, so this is the skin, human skin. Okay, so let's see what they're going to ask us. All this they give you letter, probably they ask us to identify. So we need to know the diagram of the skin and the label of the diagram of the skin. We need to know. Okay, because they will ask questions. Say, state the name of cells P. State the names of okay, you take a look at this. Take a look at this. This is a sensory neuron. You remember, right? P is a sensory neuron. Okay, so it's a look at it. This way, if you ever see the diagram of the sensory neuron, you see P is a sensory neuron. So P is question number 4A. It's a sensory neuron. Let's change the color. Sensory neuron. Sensory neuron. Okay, so that's for question 1A. Let's go to the B. Okay, 1A, Roman figure 2, state the types of blood vessels labeled QST. The type of blood vessels that are labeled Q, S, and T. Now let's take a look at them. Q, we start with Q. So the blood is going through Q, right? It's going this way to where? This is a vein. This big one is a vein. So the one joining the vein will be venous, the small venous. Venom. This is the vein. This one. You can see the white gap in the walls of this one. You can see this is an artery. Okay, this is an artery. This one. You can see the walls of the artery are thin walls. Okay, thin and thick walls, but not with a narrow gap. Thick wall with narrow gap for artery. But for vein, it's thin wall with wide gap. Okay, so this is a vein, and this one is artery. So what joins the vein within itself is a Q will be considered venum, a small unit of vein. Venum. So we say Q is venum. Venum. Okay, let's see the next one. Say Q, S. Let's look for S. S is what? Artery. This will be arteroid. The small part of artery is arteroid that is going to the capillary. It's called the arteroid. Okay, this is arteroid. So S is arteroid. Arteroid. Small segment of an artery. And then let's see T. What is T? Let's look for T. Where's T? Oh, T is capillary. These ones are capillaries. Okay. This is a vein. This is a big vein. This is an artery. This small unit is called arteroid. This small unit is called venu. And this one is capillary. Is that okay? So T is capillary. T is capillary. T is capillary. 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 Okay, let's go to 4A Roman figure 3 now. This is 4A Roman figure 2. 4A Roman figure 2. A Roman figure 2. Now we go to 4A Roman figure 3. Question 4 is a lot. 4A Roman figure 3. See, we're seeing only A. 4A Roman figure 3. Okay, let's change the color for this. Something different. Something slightly different. Okay, 4A Roman figure 3. Let's read the question. State the name of the tissue at R that provide insulation. The name of the tissue at R, let's look at R. Any insulation is fat, okay? Fatty tissue. Anything that provides insulation in our skin is fat. So fat or fatty tissue, you can write any of that. The name of a tissue that provides insulation in our body. Insulation means that it prevents other things. It's fat that provides insulation in our body, okay? In our skin. So you say fat or fatty tissue. So for Roman figure 3, we write fat or fatty tissue. Fat. Fat / fatty tissue. Fatty tissue. Fat or fatty tissue. So, okay, now let's see the next question. Let's see the next question. 4A. Okay, now we're in B. Finally, we're in B. We're in 4B. He said the blood flows through the skin of some volunteers. The blood flows through the skin of some volunteers was measured with a flow meter when their skin was exposed to different temperature. Now, capsaicin is a compound that gives people the sensation of feeling hot when it's put on the skin. Researchers applied the capsaicin to the skin of volunteers and again measured the blood flow through their skin at different temperatures. Now figure 4.2 shows the result. Figure 4.2 shows the result. Okay, with capsaicin, without capsaicin. So without capsaicin, the graph will be like this. Without capsaicin. With capsaicin, it takes a faster time to reach. Temperature of the skin is plotted. The average blood flow as a percentage of maximum blood flow is plotted against the temperature of the skin. Now let's see the question they go to ask. They use the information in figure 4.2 to describe the effect of increasing the temperature of the skin surface on blood flow to the skin without capsaicin. Okay, say use the information figure 4.2 to describe the effect of increasing the temperature, okay, the temperature of the skin surface on blood flow to the skin without capsaicin. So if we increase the temperature, what will happen? Blood flow remains constant and then increases. First, it will be constant. Look at it. To be constant. So we can see that the blood flow will remain constant, then later increases. So we can state that obvious first. We can state that obvious. So the blood flow will remain constant and then later increases. So B, 4B. Say constant, the flow. The flow remains constant and then later increases. Constant and then increases. Increases. The blood flows remain constant and then increases. That's the first part from the graph. Okay, you can just look at the graph and see what information you have. Okay, blood flow remain constant at or the values, what temperature and what's the value at of the constant. So you can measure here to here is 10, here is 2, here is 4. Okay, between 4 5. Blood flow is constant and at this, and also you can give the temperature at which it was constant. I think the temperature is around 25. Temperature is around 25. Up to the point it was constant is around 25. So we can measure this information. So constant, the value of the blood flow. Can measure the value of the blood flow first. So constant at the value of the flow rate, the flow value, 4 or 5, 4 or 5 or 4.5 or whatever, 4 or 5. You can, what's the unit of the flow? They're using percentage. Okay, 4 or 5%. They're using percentage value of the flow. So 4 or 5 at what temperature? We write the temperature at 25 degrees Celsius. Constant at the blood flow was constant at this flow rate and at this temperature, 25 degrees Celsius. Then before we now mention what is happening. So the blood starts to increase at 25 degrees Celsius, right? Beyond 25 degrees Celsius. The blood started to increase gradually, exponential increase. The blood started increasing exponentially beyond, when the temperature was now increased beyond 25, the blood started to increase. Okay? So beyond 25, so if you raise the temperature beyond 25, let's say 27 or 26, beyond 25 degrees Celsius, the blood started increasing exponentially, non-linearly. Okay, increasing exponentially, meaning it's not directly proportional. The increase is not directly proportional to the flow rate, is not directly proportional to the temperature, the amount of temperature. Exponentially or non-linearly proportional. Okay, it's better to use that word, non-linearly proportional. Okay, so that's that step. Then it will be maximum at what value and what temperature. You measure the minimum when it was constant, when it started to increase, what value of the this thing started to increase, and then what value is in maximum. The value of the flow rate is maximum at 100, the flow rate, and the temperature at that is around 40 or 40 something, 41, 42, 41. Okay, so we say the blood is maximum, the blood has a maximum flow rate, so we just, it has a maximum, so I just write maximum flow rate at 100. At 100 degrees Celsius. It has a maximum flow rate at 100 degrees Celsius. Sorry, 100%, not 100 degrees Celsius. The flow value is 100. Okay? Maximum flow rate at 100%. Okay? And then it has that temperature is 41 degrees Celsius. So you can say at a temperature of 41. So at a temperature of 41. Okay, I'm not writing those ones. At a temperature of 41. So all these points will give you this mark. All these points will give you this mark. So about 4, we eliminated 4 points, if I'm not mistaken. So let's see. We get how many marks for that question? For, let's aim for what? 4B, right? It was 4B. Okay, B Roman figure 1. How many marks? 3 marks. Okay, we've written 4 points. 3 points enough. We get 3 marks. Okay. Now explain the mechanism that increases the blood flow through the skin. The mechanism that increases the blood flow through the skin. Number 1, what increases the flow of blood through the skin? For B Roman figure 2. Mechanism that increases the blood flow through the skin. 1 is the detection by the sensory neuron. Detection by the sensory neuron. Sensory neuron. Right? The sensory neuron will sense the blood that is flowing. Okay? So that's the first point we make. Detection by the sensory neuron, the receptor organs. The receptor organ in this case is the skin, or the receptor organs. So the receptor organ is the skin. The flow of blood is detected by the receptor organs. So we can say e.g. skin. In this case, we refer it to the skin, the human skin. Okay, so that's the first point. Another point we can make under this type of question is the sensory neurons sending the impulse to the brain, right? Sensory neuron, sensory to the brain. So I will just summarize it like this: sensory to brain. And in the brain, it go to the motor neuron, right? Like the brain to motor neuron. Brain, you know, brain will interpret. Okay, brain, let's say brain interprets. And send to the motor neuron. And then I put this arrow, meaning to motor neuron. Although the motor neurons are in the brain and they will do that job, and then pass it to relay neuron. Sorry, the brain interprets are passed to the relay neuron. The relay neuron, they are the ones that are in the brain, and they will do they are part of the interpretation process, relay neuron, and then the relay will pass to the motor neuron. Then the motor neuron will bring the message to the effector organ. Okay, so relay to motor, relay to motor neuron, relay to motor neuron, then motor neuron will bring to the effector organ. Motor to effector organ. Okay. Always mention impulse, because I was abbreviating, so I didn't write everything. Motor to motor neuron. You see, I abbreviated motor neuron. I just wrote motor. So I'm not writing all the words here, but I'm just telling you that I'm mentioning them. You say electrical impulse, you always mention something like that. So motor to what? The effector. We have the receptor, we have the effector. The effector is the one that will not eventually tell you what has happened. The effector organ, which is the skin. Effector organ. So if you can get at all these points, then you will get your desirous mark. You can also mention some of the things like the arterial and the vein, which are parts, because we are talking about blood. Okay, you can mention the muscles in shunt vessels, they contract, and the arteries, they relax. Muscles contract, the arteries, they relax. The dilation, you can mention dilation of vasodilation that's taking place, which increase more blood flow in the capillary. So we can measure all of this. Measure dilation, contraction of the muscles. Measure dilation. Before dilation, you can measure the contraction of the muscles. When muscle contract, the artery will relax. Contraction. Of muscles. We measured dilation, vasodilation also. Vasodilation. We mean to write vasodilation. As is taking place. Then this will eventually increase the blood flow. This will increase the blood flow. Increase blood flow. Okay, so this for this question. Let's go the other question so that we can finish question. This one has about 3 marks also, right? 3 marks. Now we go to the Roman figure 3. It says state the difference between the average blood 4 Roman figure 3. State the difference between the average blood flows for the treatment with or without capsaicin at 35 degrees Celsius. So we have to go to the graph at 35 degrees Celsius. Look at the temperature at 35, trace it to the line, to both of them. If you trace 35 here, trace it here, the value is without capsaicin is 20. Okay, continue to trace it to meet with capsaicin. Continue to trace it up to meet capsaicin, and you minus the values, subtract the values. Here is 60 what? Seems like 65, right? Am I right? Or 66? So 65 or 66. So 66 minus 20. So that's what you're going to do to find the difference, because it says we should state the difference, right? We should state the difference. Question B Roman figure 3. 4B Roman figure 3. Roman figure 3. Okay, the difference is, with capsaicin, the value of the blood flow in percentage is 66 is what we found just now, right? The value is 66% with capsaicin. So I just write with C. C means capsaicin. Capsaicin. With capsaicin, we get 66%. If you trace from 35, then without is what? Without capsaicin, without capsaicin, the value is 20. If you trace from 20%. Now they say we should find the difference. The difference is with, which is 66, minus without, which is 20. So this will give us 46. This will give us 46%. So we just write here 46% as our answer. 46%. Okay, we write 46. So this is our answer for this question. 46%. So that's what we get. 46%. B Roman figure 3, right? Okay, Roman figure 4. B Roman figure 4. It says the researchers thought that capsaicin stimulated receptors in the skin. Explain the process by which the capsaicin could reach these receptors. The process, first you state it and then you explain. The process is through diffusion. Question 4B Roman figure 4. Question 4B Roman figure 4. The process is through diffusion. So we write diffusion. You know diffusion, right? The movement of gas molecule from region of high concentration to region of lower concentration. So you just write diffusion and you begin to explain how. Why is it this time, why are we calling it gas molecule? Because it's the temperature that has made it not just ordinary liquid. Okay, so we say down concentration gradient, high to low. That's from high concentration to low concentration. So you write it, you know the diffusion. So the movement of gas molecule gas, or sometimes even liquid molecule, but usually gas molecules from high region of higher concentration to higher concentration to region of lower concentration. So region of higher concentration to region of low concentration. So I just write high to low. The concentration you already know. To low, okay? Conc. Okay, conc means concentration. So from high concentration to low concentration. So that's diffusion in this regard. And then in this case, we can measure also active transport because you require energy, right, to move it. So we can say active transport. We can also use active transport, measure active transport, which requires some energy to move it. The energy could be the oxygen that helps it to move. Okay, so active transport. Through what? Through the epidermis, the skin, through the epidermis layer of the skin. Through the epidermis layer. Through the epidermis. Okay. Each of these points will give you marks. You can say through the cell or into the cell or through the cell. And then across the cell membrane. So across, through the cell or across the cell membrane. So when we write all these points, we get our marks across the cell membrane. You must use the biology terms in order to get the marks if you are doing biology. If you are doing chemistry, you must use the chemistry terminology in order to get the marks. If you are doing physics, you must use the physics terminology in order to get the marks. Okay? So these are all the points, and this will give us the marks that this question is desirous of. Now let's move to the next one. This one you also get another 3 marks. Now we are in question C. Explain the importance of regulating body temperature in humans. Why do we regulate body temperature? One of the reasons is that, there are enzymes in our body that helps in the digestion process. If we do not regulate temperatures in our body, the enzymes can become denatured. The high temperature can denature the enzymes in our body, right? One of the reasons why we have to regulate our body temperature to prevent the enzyme from being denatured. So I'll write enzymes, I'll write denature. Okay, I will not write the full sentence. Enzyme, not, I use the word not, denature. Because when they denature, they are destroyed. The enzymes can no longer break down our food, right? The active site will no longer feed the substrate when they are denatured. The enzymes will no longer feed the substrate and will no longer be able to break down our food. So we have to regulate the body temperature and so that enzyme remain active and maintain a optimal temperature for usage. Okay, so that the enzyme becomes remain active, so they can remain active. To remain the enzyme. When I say to remain, I'm talking about the enzymes. To remain active. Remain active. If you don't regulate the body temperature gets too high, it will destroy all the enzymes, the digestive enzymes, and then we will not be able to break down the food we eat, and then we're in trouble, and then we die quickly. So to remain active. So the enzymes continuously has to be active in order to be able to carry out their functions. Okay, and too much temperature will destroy them. Okay, that's the first part. So to remain active and work at their optimum temperature. Active to work at their at their optimum. You know, optimum is like the desirous maximum temperature at their optimum or best temperature for the system. Optimum temperature. To work at their optimum temperature. To work at their optimum temperature. So that's one thing to note. Another thing we can also say is how many marks? It depends on the number of marks. 4 marks. Okay. Another thing we can also say is to avoid we have to maintain the temperature so that the metabolic reaction in our body will be at their constant level. Okay, so a point on body metabolism. You know, the metabolism like respiration, digestion, all the activities, chemical activities taking place in our body, they are metabolic activities. So to keep the metabolic activities constant, metabolic activities constant. So one reason we maintain constant body temperature is because so that we'll be able to keep the body metabolic activities constant. Okay, because everything, the digestion has to be at the rate it should be, the respiration has to be, excretion, all the body metabolism must be at the desirous time and desirous temperature. So we keep body temperature constant in order to achieve all of this. Also to avoid damage to our proteins. You know, protein is the life. Protein is the life. If no protein makes the enzyme, protein makes all the insulins, pancreas, all these things. So if your proteins are damaged, then you are damaged completely. So to avoid damage to the body protein, to avoid damage, so you have to keep the temperature of the body constant to avoid damage to the protein. To protein synthesis or protein itself. Okay, protein is life. Protein makes the DNA. Protein makes the gene in the DNA. Protein makes the gene in the DNA. So if anything happens to your protein, you are in trouble. Protein is responsible for everything, most of the things in our body. Also to avoid damage to cell membranes. If you don't keep the constant body temperature, you can damage your, to avoid, okay, the avoid, I did write avoid, to avoid damage to cell membrane. If your cell membrane is damaged, then there's a problem, right? To avoid damage to cell membrane. This homeostasis. This is homeostasis, okay? They are asking you question in homeostasis. Why do we have to keep the body temperature constant? If the temperature is not constant, it can affect the enzymes, can denature them. If it's too high, the temperature is too high, it can damage the enzyme and they will not be able to work at, they will not be active and they will not be able to work at their optimum temperature. It will damage the metabolic activities and they will not be happening at constant rate that they are supposed to happen, like the digestion, respiration, circulations of blood in your body. All these are metabolic activities in the body, and then we keep this thing constant to avoid damage to protein, and so to avoid damage to the membrane. And the heart, to avoid like heart attack, stroke, hyperthermia, overheating of the body is very important. This one we need to state. to avoid overheating, body overheating. You know the way car overheat? The body can overheat. To avoid body overheating. Okay? You see overheating, the way it's dangerous to car. When our body is overheating, it's very dangerous. Avoid body overheating. So we need to put this point down. There are many points. Body overheating. To avoid heart attack, to avoid stroke, to avoid so many issues. Okay, so you have to keep the body temperature constant. Okay, so that's that for that. Let's go to another question, because I think this is 4 marks, and I think we have already been able to attract the whole 4 marks. Let's go to D. So I think this question is 22 marks. You can see how it's very long. Why it's very long? Okay, so we go to question D, question 4D. Let's quickly answer this, and then we call it a day for today, for part 2 of this. Now, body temperature is controlled by both hormones and nerves. Explain how coordinations by hormones differ from coordination by nerves. Okay, hormones, is in the endocrine system, right? And then the coordination by nerves is in the nervous system. Okay, so hormones, they are chemical substances. Okay, they only used for chemical coordination. They are chemical substances. While the nerves are different, right? So hormones are chemical substances that helps in the chemical coordination in the body, right? And they are transported by the blood in the circulatory system. Okay? Hormones, question 4D. Hormones are what? Chemical substances secreted by what? Hormones are chemical substances chemical substances secreted by glands, right? Secreted by glands. By glands. Into the blood. So the blood carries it to every organ that it needs to. When it gets to the organ where it needs to act, it drops down to the organ, into the blood. So you can measure that they move in together with the circulatory system. So here you measure transport system or circulatory system. Helps to move hormones. Circulatory system, because they move in the blood. Circulatory system to the desired organs, the specific organs. To the specific organs where they need to act, right? Specific organs. To the specific organs where they need to act. Or you can say targeted organs. If you don't want to use specific, you can use targeted organs. The word targeted, I think it's more appropriate. But specific is also okay. meaning not every organs. Only the organs where they need to act upon, that's where they drop down from the blood to the organs. So specific or targeted organs. Okay? Targeted organs or specific organs. Okay, so that's for hormones, because they want us to tell the difference between hormones and the other one, the nerves. So they are, you can also say they're slower than, it's a slow process. They're slower than nerves. Okay? Slow process. Slow. Hormonal system is a slow process, or slower than nerves. Yes, slower. It takes quite some time for the hormones to act. Then they are slower than nerves. Nerves is a fast response, fast process, right? So hormones are slow process, slow response, not as fast as this, okay? And they take longer and they are more lasting. Take longer time and they are more lasting. Hormones, they take longer time, and they are more lasting. And are more lasting. When we mention that these ones, you can also benchmark with, say these ones are dropped into the circulatory system. You can mention here why the nerves are in the nervous system. You know, like every point you give the why nerves are in nervous system. Nervous system. Here we measure hormones are slower than nerves. See? Slower. I did write the hormone, but you should write hormones are slower than the nerve cells. Hormones take, you understand, or hormones takes longer time than the nerves. And the hormones are more lasting. This is another point you can bring down here. Okay, so these are the points you need to write in order to get your marks for this question. So I think part 2 will be stopping part 2 today. Part 3, we will be taking question 5 and 6. Okay, thank you for joining us. Hope to see you again in our next class.